The speaker for today is old and mean and ugly and me. And I'm going to talk about something that is a bit of a departure from what we've done in the past, where we have been looking at research programs sort of department by department. This group of investigators I'm going to talk about now, we're not in a department per se, but we're in an institute which actually has a lot to teach us about how science can and should be done at the research level. It's the Institute for Enzyme Research, a building that still exists but stands empty now on Old University Avenue. So if you go up University Avenue to where it splits with Campus Drive, there's a tall wedge-shaped building, which is the energy building, new energy building. Then there's the ROTC building, and then there's the Enzyme Institute. At one time, that was one of the most prestigious places in the country to do science, and it's sad that didn't last. I'm going to tell you about what it did do, and then we can figure out why it didn't last. To tell you about the place, I have to tell you about some of the people, and some of them are people you have already heard about, as is the case for Conrad Elvium. Elvium grew up within sight of the campus in McFarland, got his PhD on campus here with E.B. Hart, which was very common then. As a student, there were 3 or 4 students at Hart's laboratory at that time. And then, as was very common at that time, he went to Europe. In his case, it was the United Kingdom and the laboratory of Kalin. But this was very common for American postdocs. They'd finish a PhD here, they almost always went to Europe. Kalin was in Cambridge, but there were people in Göttingen and Tübingen and Berlin, all of whom were sort of finishing schools for American PhD students. And in fact, during that period, say from 1900 until 1940, science, and especially chemistry, was basically a German science. That's where people went to get the straight dope. Elvium, after coming back here from a year or 2 with Kalin in the United Kingdom, is the one who discovered pellagra to be curable by niacin. You remember I told you about this a while back. He did a number of other things too, and I'm not here to tell you about them so much as just to introduce him. But he was an enzymologist of the first rank. He was a member elected to the National Academy of Sciences. He won the Lasker Award, which is very commonly considered the last step before winning the Nobel Prize. And I have seen the Nobel nominations for him. He was nominated 12 times for the Nobel Prize. After some time as the chairman of biochemistry, which by that time was a big department, so the chairman had a big job, he took over the deanship of the graduate school, which of course is an even bigger job, and shortly thereafter became the president of the UW, where he served until he died literally in office. He keeled over and died in his Bascom office. I'm telling you about him now because he was one of the 2 people who really moved the campus towards having an enzyme institute. The other one was Van Potter. He was 10 years younger than Elvium, and in fact had been Elvium's PhD student, so he got his degree training with Elvium. As an aside, he actually got his undergraduate work at South Dakota State College, and there was a group of 6 people, all from South Dakota State, all the same age, all of whom came here, and all of whom had distinguished careers. Another one that I've talked to you about, I think, is Bob Burris, who was a member of the National Academy from the same, not only from the same university, from the same laboratory there. One person sent these people here. So, as was common, he did his postdoctoral work in Sweden with von Euler and in the United Kingdom with Krebs, who had moved by that time to the MRC laboratory in Cambridge. And he joined the faculty here in 1940 and stayed for 61 years. He was also an elected member of the National Academy and very, very widely regarded as a good scientist. He also was a living, breathing human being who cared a lot about stuff outside the laboratory. And he introduced the idea, in fact, I think he introduced the term global bioethics by writing a book about it well before other people were thinking along these same lines. It's a book that deserves to be read now. It ends with a sort of creed for the person who cares about the earth, and every word of it is still perfectly applicable today. So Van Potter and Elvium, who was his senior, went to a meeting someplace in the East Coast and shared a room. And as they were falling asleep, one of them said, "You know, we really ought to have an enzyme institute." And the other one said, "Yeah, we should." And I'll tell you a little more about why they said that. But anyway, they got the idea sort of out of the blue. But it made lots of sense. At that time, the Kaiser Wilhelm Institutes in Europe, in Germany, were the top places to get trained. And here, for example, is the Kaiser Wilhelm Institute for chemistry and for electrochemistry and physical chemistry. These were buildings that were paid for with the government, supported by the public. They housed powerhouse teams in specific areas, and they were organized in a way unlike anything we're familiar with in the university. There were no students, for one thing. Each of these institutes had a boss, and he was king. The boss here is not hard to recognize. He's, of course, the only one allowed to wear a beard. But here you see his crew around him. They are, of course, all men. At that time, there were absolutely no women allowed into this club. And the boss here, and I'm afraid I don't even know which lab this is, so I don't know who the boss is, would come in the morning, say, "All right, team, this is what we're going to do today. You do this, you do that." They were essentially a well-functioning team, all of whose efforts reflected well on the boss. And so the boss went out and spoke about their work. He got credit for it. He very often won the Nobel Prize for it. But the younger men there, and later women, got the experience that was sometimes exceptional. I mean, one of the reasons that experience is exceptional is that those institutes were headed by people who were exceptional. This list is all the people who were they're all people who were heads at one of the Max Planck Institutes, and every single one of them is a Nobel laureate. Some of them you've heard of course, Heisenberg. Warburg at that time was the world's most prominent biochemist. Haber and Hahn were German chemists that were famous and became more famous after their service in this place. But the idea was that you hired somebody who was absolutely world class. You brought him into this place. You supplied him with everything you needed to do research, then money, space, help. You loaded the lab with young scientists still in training who are willing to put up with lousy conditions and low incomes. And if you were lucky, you were in the laboratory when something great was discovered, and you had the reflected light of a Nobel laureate to live under. The other feature of these things was that when the boss died, the whole thing dissolved, and they started all over again. So if you cared at all about having a job, you tried to take good care of your boss, I suppose. But the nature of this was that when somebody died, that whole line of research was discarded. Somebody new was hired, and one of the advantages of that was that it was an automatic method of updating the research in a place on a scale that was about a human life scale. So these places stayed caught up. The Rockefeller Foundation at that time had lots and lots of money, which they spent for lots of things, including health-related issues around the world. But one of the other things they did was to fund postdoctoral studies. And these people that I've talked about here, Elvium, who else did I say? Elvium, Van Potter, they were both Rockefeller Foundation fellows. It was a highly cherished award. I mean, it really meant a lot. If you've read Aerosmith, you may have heard about the Rockefeller Foundation. In any case, it was the pinnacle of success in science to be one of their fellows. This will seem like a digression, but in fact there's a reason for my doing this, so stay with me. Otto Warburg was the biochemist that I said was the the leading biochemist of the first half of the 20th century. He isolated all of the enzymes that are involved in the process of converting sugar in your diet into CO2. He discovered in the course of studying those enzymes that the enzymes required other structures, coenzymes, low molecular weight things that turns out were derived from vitamins, as we've talked about, and which are essential to the action of the enzymes. He recognized early on that iron played a special role in biology, and he didn't understand what that role was, except in very broad terms, but that was an important step forward. And he made one observation that still is at the very top of the list of questions being investigated now, and that was the observation that tumors of all kinds, when they grow in the laboratory or in the individual, use a method of getting energy from glucose that is inefficient. The method is called glycolysis, and to give you a rough idea of how inefficient it is, you get 18 times more ATP if you use the alternative, that is burning the fuel in oxygen, and nonetheless tumors prefer somehow to burn carbon without the use of oxygen in a much less efficient way. And it's the so-called Warburg effect, and it's still being investigated. There's some slight understanding of it now, but he was really way out ahead in the work he did. And part of the reason was that he had a place, the Max Planck Institute, a technician, Christian was his name, stayed with Warburg for many years. A method, here it is drawn for you, the respiratory measurement of enzyme reactions. I don't know whether you can see enough of this to make sense of it, but of course, in those times when he started his work, there were no spectrophotometers that worked in the ultraviolet. So that a lot of biological molecules that are absorbent in the UV couldn't be followed, so you needed something else. And what he did was to measure either the release of CO2 when a substrate like glucose was broken down, or the disappearance of oxygen when oxygen was used to oxidize something. And the equipment for that was a flask, a small flask here. You could easily hold it in the palm of your hand. The flask was tightly fitted to a joint that brought it to a manometer. And the glass was also built with several little side pockets that could be used to tip solutions into the main part of the flask to start a reaction. So you set everything all up, you close the system so that there's no leaking of air in any place. You tipped in the stuff you're going to study, and then over time you watched either the appearance of CO2 or the disappearance of oxygen by reading this manometer as it changed gas pressure. It was an amazingly effective device, and with it he did absolutely amazing things, especially when you think of how little he had to go on. I mean, there simply was very little biochemical groundwork for him to go from. Warburg won the Nobel Prize in 1931, and he probably was nominated every year from 1920 until 1950. Barkhoff was another person who developed an alternative to the Warburg manometer. It was called the Barkhoff respirometer, but it basically followed the same principle. You measured disappearance or appearance of gases. Elvium, when he was in Europe doing his postdoctoral work, wrote back to Hart, who was then the chairman of this department. I was glad to have your letter and to learn that there would be a job waiting for me when I returned. Remember, I told you that in this department and in most departments, the way they staffed was they hired their best current graduate student. And then when that graduate student got old, he hired the best graduate student around. So there are generations of students, and Elvium and Hart was one such example. So he wrote back saying he wasn't crazy about the amount of money he was being offered, but he really wanted to have a Barkhoff respirometer, a device like the one that I showed you in the last slide, some slight variations. And Hart did buy that for him, and he brought it back here and did lots and lots and lots of experiments over a period from, let's see, it would have been from 1930, I think, to his death in 1961. So he was a very productive scientist, and it was important science. It wasn't just hack work. I have that Barcroft respirometer, and someday I'm going to reassemble it in a museum, and you'll be able to see what he worked with. You'll be astounded to see what simple tools they had. Well, so this is the way things were up until 1940 or 1939. The Kaiser Wilhelm Institutes all over Germany were the premier place to get trained. But of course, everything changed with the war. And this example is the chemistry Max Planck Institute, or no, sorry, Kaiser Wilhelm Institute. They came to be called Max Planck Institutes later. So the Kaiser Wilhelm Institute in Berlin on the left, and after the war on the right. And this was typical of all these scientific ventures. They were just absolutely blown to ribbons. And there was therefore no place in 1945 or 46 or 47 for an American postdoc to go and get the finishing that postdocs used to get. And it was in that context that Elvium and Potter talked over the need for an institute like that at Wisconsin. And by like that, I mean these things. Very well supported, separate from the university so that there are not undergraduates to teach, long-term funding so you don't have to be running around after money all the time, people that were long-term associates and excellent scientists, and the process of wiping everything out when the boss died. That's what they were thinking about, at least everything except that last, probably. So here are three people, Potter, Elvium, and King. King was at this time in the government, in the federal government agency that funded science. He also was the guy who almost won the Nobel Prize for discovering vitamin C. Should have won it. But King, having talked to Elvium and Rand Potter here, suggested that the Rockefeller Institution should look into the possibility of a Kaiser Wilhelm-like institute. Potter and Elvium said, if you're going to build any institute, let it be for enzymology and let it be in Madison. E.B. Fred, whom we have, we have not talked about him, but he was a famous scientist here, bacteriologist, eventually became president of the university. Fred liked the idea and put up some money. By this time, Wharf, about which we have talked, was making a lot of money on its patents, the Steenbock's patents, for example, and they were able, therefore, to kick in a couple $100,000 to match $100,000 from the Rockefeller, and the Enzyme Institute was built for $300,000. That's amazing, but of course, those are $300,000 in 1945 or so, and it probably was at least 10 times more money than what it looks like there. But it did, in fact, result in a new place, different from anything else that was on this campus. And of course, the needed next step was to find the people who would be the bosses. David Green, who was born in 1910 and lived until long after I came here, so I got a chance to know him well, was the first one selected for this institute. And he was already quite famous by the time that they set up the institute. He had worked in Cambridge with a famous enzymologist, Malcolm Dixon, for 8 years. He was born in New York City, and he had the accent that goes with the New York City product until he went to Cambridge. When he came back from Cambridge, he had acquired a cane and a British accent, which lasted the rest of his life. Very interesting guy. But he was a superb enzymologist, and he set up his space in the new Enzyme Institute building. And his interest was primarily in understanding how humans, but other animals as well, broke down fats for energy or made fats to store energy. And if you page through a biochemistry textbook now in the chapter about fatty acids, you could just about point to any page and there'd be something there that David contributed to. He was a very productive enzymologist. He was also a tough guy to get along with, and he ruffled a lot of feathers in the first couple of years he was here, and it became clear to the people who had started the Enzyme Institute that they had to find somebody to counterbalance him and keep him from going off the deep end. He was just a little bit egotistical. Here's the person they chose, and they chose very, very well. Henry Lardy is the person I really wanted to tell you about today. He, like Van Potter, was born in South Dakota and went to the University to South Dakota State, worked with the same man there as an undergraduate, came here in 1941. He came to biochemistry and got his PhD in this department, working with Phillips, who was also a widely regarded biochemist at that time. And then he also went abroad. I'll show you who he studied with abroad. Later in his life, he won the Wolf Foundation Award in agriculture, which is the Nobel Prize in agriculture. And while he was here, he convinced everybody around him that it was possible to be a good biochemist and also a decent human being. When he was an undergraduate, he worked with this guy, Alvin Moxon, and part of what they were interested in were the effects of selenium. Selenium is toxic to cattle, to dairy cattle, and there are places where selenium is rich in the soil and therefore gets enriched in the forage, and cows get sick from it. Larney tells a great story. If I had time, I'd read it to you, but it was about the experiment in which they fed dogs sublethal levels of selenium, and one of the effects was that their kidneys just quit working. And Lardy had the idea, there was good reason for it, which I can't take the time to explain, but he had the idea that a compound called bromobenzene might actually reverse the effects of selenium. And he describes how they set up a series of dogs, left them overnight with bromobenzene in their food, and they came back the next morning and found all these dogs knee-deep in urine because their kidneys had suddenly kicked in and they were making urine like crazy. So they actually discovered in his early undergraduate years an interesting result which later turned out to be useful in human medicine. In 1938, Moxon, the teacher at South Dakota State, took 5 of his students, piled them in a car, and they drove from South Dakota State to Madison. On the way, they stopped in Rochester, Minnesota, where the Mayo Clinic at that time was young. And Kendall, who won the Nobel Prize for work on steroid hormones, was there at Minnesota, at the Mayo Clinic, rather, at that time, stirring huge vats full of muscle and liver with a canoe paddle as the starting material for discovering or purifying steroid hormones. When they got here, they were impressed with the place, and all 5 of these students eventually came to Wisconsin. And one of them was Madden, that I think I told you about in connection with the discovery of niacin. Another was Weissman, who is the one whose name is on the Weissman Center, and who did the early work on phenylketonuria. Here's Lardy on the left, sitting in the back steps of biochemistry right just across the street. That's Paul Phillips in the middle, and I don't know who the other one is, clearly somebody in that group. But Lardy looks like a schoolboy. He had just arrived here, and he spent a long time here. There's a time later I'll show you how long it was. His first assignment was daunting. His advisor, Paul Phillips, had done work on artificial insemination of dairy cows, and they had reached the point where they were very successful at artificially inseminating one cow. But to be really effective, what they needed was some way for bull sperm to be stable enough so you could inseminate several cows in the same farm, or better yet, you could actually ship it around the country and around the world to breed better dairy cattle. But the problem was that bull sperm was not stable as collected. So Lardy was given this bull. He called him Pap's Comet, a vicious beast, is what he described. And his assignment was to figure out a way to stabilize sperm with stuff that was real easy to get on a farm. So it couldn't be exotic and it couldn't be expensive. It had to be simple. And it turned out that Lardy, after some smart thinking and some hard work, found within 6 months that egg yolks, supplemented with a buffer to keep the pH constant, was the ticket. It stabilized sperm for 10 days, which was a huge, huge improvement of the situation. It put Wisconsin on the map for artificial insemination. It was the beginning of a huge industry, which is centered around Madison now. If you drive around Madison on the north side of Highway 90, There's a place there, what's it called, ABS? Yes. ABS, which is a place where they export outstanding bull sperm all over the world. It's a multi-billion-dollar industry now. This is how you collect bull sperm. Sometimes it's dangerous. This is an article from the paper that describes this first work, and here, this is Paul Phillips, that's Lardy. These guys are standing around an airplane, and the reason for that is that even with sperm that would last for several days, it was very hard to get the bull sperm to the farm when the cows were in heat. And if you didn't hit the window, then you were out of luck. It wouldn't work. And in a good share of the season in those years, back in the 20s and 30s and 40s, country roads were impassable when they got muddy. The agricultural agents used to go around on motorcycles, and they even couldn't get through some of these muddy things. So, this is the truth now, what I'm about to tell you. They delivered bull sperm by parachute. And this is actually a bull sperm parachute that's in the Historical Museum in Madison. This process was much improved over the years, and one of the things that led to improvement was the discovery that if you just lowered the temperature, then bull sperm was stable almost forever. And now these enormously productive herds in Wisconsin, but elsewhere all over the world, are often herds that were derived from a few prize bulls that were raised here. If we had time, I'd like to tell you another story about this, but the whole business of reproductive physiology, artificial insemination, in vitro fertilization, much of that was done here. The early work on in vitro fertilization came from here. So Cassida was the one who started this work in the 1930s, but this was before the preservation of sperm was possible. Ed Heiser was the Department of Dairy Husbandry's chairman, and he sort of set up the long-term opportunities for studies of artificial insemination. They produced, as I say here, a film on artificial insemination, which was widely used all over the world. They found that farmers needed to talk to each other about how best to use this powerful technique, and they set up animal breeders' cooperatives. So altogether, it was a major addition to the armor of the weapons of farmers. Lardy did this in 1941, 6 months after he became a graduate student. And let's see, I think it was 45 years later he won this prize, the Nobel Prize in Agriculture, the equivalent of the Nobel Prize. It's a long wait for the thing, but much deserved. So we go back to where we started now with the Enzyme Institute opened. Lardy was there. David Green was there. Lardy had come there after being a postdoc at the Banting Institute in Toronto. Banting was the one who with best discovered insulin. And while Lardy was with Banting, at the Banting Institute, he studied with Fisher, the son of the very famous carbohydrate Fisher. While he was there, he learned to synthesize in the laboratory some sugar phosphates, and that, because those sugar phosphates turned out to be important in metabolism, the way we break down sugars, for example, he became interested in metabolism, and that was the rest of his life, practically. When he came back here, the building across the street looked like this. All those extra $10 million worth of buildings, $100 million worth of buildings weren't here yet. And the College of Agriculture still was very much aimed at solving practical agricultural problems. And among the things that Lardy did was to find, this was a surprise to me, but apparently he was able to discover a way to detect DDT in crops and got into the butterfat of dairy cows. And he was among the first people to contribute the idea that we could have trouble from this DDT. And I'm trying to think of who wrote Silent Spring? Rachel Carson. In her book, she actually notes that Wisconsin scientists were among the people who first noticed the effect of DDT on bird eggs, and so some of her conclusions came from data from here. Lardy worked with a student of Albium's. The two of them figured out, made a major discovery in science, one that would have made him famous by itself. They discovered that a certain vitamin, biotin, was critical in a large series of processes in which carbon dioxide from the air was fixed into things we could use, and that was done while he was still a very young man. Lardy didn't focus on one thing. He had a whole bunch of things going all at the same time. I guess he had a lot of people around him. So he didn't have that laser focus that sometimes leads to a Nobel Prize, but instead he had his hands in a whole bunch of things, and everybody who knew him admired him for this virtuosity. So folic acid is a vitamin, serine is an amino acid, as is glycine. Purines are the components of nucleic acids, and he studied the metabolism of all of them, made contributions in each case. And one of the things he wrote as an old man, looking back over his life, was that our biochemical interests were Catholic, and one's journal reading covered broader areas in the 1940s than is possible now. This was written, I think, about 2004 or 5, and if anything, it's even truer now that science has gotten so deep that you can't be broad anymore. I mean, it's very hard to be broad, and one of the losses is that the kinds of collaborations across departmental lines that used to be common here don't take place nearly so often. Here's a quotation from Lardy, which, I mean, coming from him, it's not so surprising, but from most people it would have been amazing. As a graduate student, I shared authorship with members of 5 other departments. That's the way they worked then. So what did he do? Well, he carried on his interest in sperm by studying with this Warburg respirometer that I described for you, studying how they generate energy, and it turns out that they generate energy in a way different from most of the rest of our cells. He looked at thyroid hormone, had some early understanding of how it affected metabolism, speeds it up. He discovered the effect of a compound called dinitrophenol, which, when it hits mitochondria, makes them function very inefficiently, so that instead of turning up producing ATP, they make heat. And this was his entry into this whole field of thermogenesis, into which he put many years of his life. The way that animals keep themselves warm. And again, he was so far out ahead that the work that he published in the 1950s, 60s, was practically lost in the literature before somebody else came along and repeated things and claimed that they discovered what Lardy had already done long before. It's now clear that we have 2 different kinds of fat. One that's essentially for storage of energy when we need it, and the other is for thermogenesis, to make us warm when we need it. And when we use our fuels to drive heat, that's a way to burn fuel without making ATP, and so it's essentially a way to spin your wheels and get rid of fat. When you go into a situation where you're freezing, the other fat, the brown fat, takes over and generates heat. And Lardy was already onto that 20 years before anybody else was. DNP was a popular hot item in the 1940s and early 50s because its effect on mitochondria would predict that if you ate dinitrophenol, your mitochondria would just spin their wheels, burn fat, make you thinner. And sure enough, it was a great dietary aid, whose only real defect was that it killed you if you tried it long enough. And so it never quite made it among the medicines. This is another aspect of Lardy's life. He didn't just use other people's stuff, but he designed and built apparatus that was perfectly fitted for what he needed. This is the so-called manometric microapparatus. And here's the the prototype for that machine, crazy looking old bucket, but it was his first try, and this is sitting on a bench in my laboratory. We still have that old thing. Well, so he was the counterbalance to David Green at the Enzyme Institute. He became the director of Section 2, developed a very large group of people around him, and did amazing work for many years. But it was always in the context of real struggles with David Green. There was a certain amount of space which they struggled over. There was a certain amount of direction that they would take the institute, and they didn't agree on that. And this picture of all of them smiling, this is Lardy, this is Elvium, this is David Green, is probably posed. I don't think I never saw them smiling much in each other's presence. So, just an aside, why would the Rockefeller Foundation, which founded the Enzyme Institute, choose Wisconsin? And this is a list of the people at Wisconsin who won national awards each year. It was originally called the Paul Lewis Award, and then it became the Pfizer Award. It's awarded to the outstanding enzymologist in the country. And look here, these are all Wisconsin people: Green, Potter, Leninger, Lardy, Boyer, and so on. They all won the Paul Lewis Award, and it was indicative of the fact that Wisconsin was a really major center of enzyme research. I think I've already said something about each of these things, but let me just recap. Sperm, biotin, thyroid, coupling of mitochondria and their uncoupling by nitrophenol. Gluconeogenesis is the process by which we take things that are metabolically not useful and turn them into glucose, and he studied much of that process. In the course of doing so, he discovered something that suggested that sudden infant death syndrome might be related to gluconeogenesis. I haven't followed that, and I don't know whether it turned out to be the case or not. I've told you about the cold adaptation from brown fat, and I guess I didn't say explicitly, but the idea now is that if you could get brown fat to multiply, so you have more of it, you would have a way of dealing with obesity because it burns fat. Then, in his old age, Henry, who worked until he was 90 years old or a little more, took on a completely new project. And well, I guess before I tell you about that project, let me show you a few things about him. This may not be legible to you because it's a copy of a copy, but it was hanging on his wall when I went through his office a few years ago. Working time which is lost while preparing for examination should be deducted from your vacation allotment. Anyone qualifying for advanced degrees should be able to acquire scientific knowledge during class periods and leisurely evening reading. Knowledge gained while rereading class notes is either false or short-lived. What you learn at the laboratory bench is yours forever and may even benefit the rest of mankind. I think that's rich. Here's a piece of paper I found on Henry's desk after he died. And there are two things about it that are interesting. The title has been altered to change from electron transfer, that's mitochondria, metabolic efficiency, that's brown fat, and thermoregulation he added. This is a late part of his career. The other thing that's interesting about this, I'm sure you can't read it, but I will read it for you. He wrote this fairly late in his life, and it starts out, 62 years ago, one of us found that the oxidation of ketone bodies, 62 years ago, and I look at that and I think, how many biochemists have ever lived long enough to say that they did something 62 years ago? Well, in his late years, this is really, I guess, the last 10 or 15 years, 15, he said there's not enough space in the Enzyme Institute. He was getting crowded because we were hiring new people at the Institute. And he said, with so restricted a space, I can't do my usual stuff, so I'm going to work on steroids. I knew nothing, next to nothing about steroids, but he was interested in this. DHEA, dihydroepiandrosterone, is a compound that is in the blood of humans, both men and women, and the amount in the blood drops with age. It is a precursor to both androgens and estrogens, And it's by far the most abundant steroid in the human body, but hardly anything was known about it when he started this work. This is a 25 milligrams of a hormone is a tremendous amount. So it's a lot of it, and surprisingly undisturbed by research. So he did. He set up a laboratory. He learned how to do all kinds of chemical synthesis of these things and separation of them. They followed the fate of the compounds in all kinds of animals. He tested, one day I saw him testing the ability of rats to learn to swim, and a huge range of things that he did. And he did it all with his own hands. It was he and two postdocs that worked with him, and they covered all these things. This slide is colorful because it's his, and I guess he felt that each of these things was a separate thing that needed to be emphasized. He was a good citizen, too, and I said he was a good man. Oh, can you read this?
Yes.
He didn't pull his punches, and he was very much involved in liberal politics here. He bred golden retrievers, and among the things I found in his office when I went through it after his death was a long correspondence with somebody in Minneapolis that had also raised golden retrievers, and one dog had come from one of them to the other, and they all both loved the dog and wrote back and forth about it. And he raised stallions, Arabian stallions. In the course of going through the stuff in his office, of which there was a lot, I eventually sent 50 archive boxes to the archives, I came across something that really disappointed me, and it was some poetry that I'm afraid you will not quite be able to read, but there are two versions of a poem here. And all that you really need to know about them is that one of them is signed Hal Bauer, and the other Henry Lardy. And they're clearly the same, not quite identically. There are a few little changes here. But I look at that and I said, Henry Lardy would never plagiarize poetry. It's inconceivable that he would. And I set this aside and I went on marking boxes. Henry Lardy, Henry H-A-L, H-A-L, all these boxes. Then one day it dawned on me, H-A-L, Der Bauer, Hal Der Bauer, Henry Lardy, the farmer. And so he was actually writing under an assumed name. And I found other things, short stories and such. When I talked about this at a seminar years ago in this auditorium, his wife and kids were here, and they had never heard of this either. That was news to them. He was one of a kind, and those of us who knew him were lucky. So that's the story of Henry Lardy. The Enzyme Institute has another additional 10 minutes worth of story. First, Paul Boyer, who was Henry Lardy's best friend. The two of them came here at the same time, stayed best friends for their whole lives. I have correspondence between them in their 90s. Paul is still alive, and Paul won the Nobel Prize for discovering how cells make ATP. And it was, I think, partly because he had this focus. He looked at just one thing for his whole life. If it was off to the sides, he didn't get pulled out. And the result was terrific learning. Helmut Beinert was director of Section 3 at the Enzyme Institute. He's a wonderful man who was German, who was actually into classical Shakespearean acting in Germany. Then he somehow got involved in science. Guess he didn't like acting all that well, didn't enjoy it. And then the war came, and he somehow got brought here by the group of American operatives who were bringing back scientists. He was originally placed in a kind of a useless lab in Texas, but his brilliance showed, and eventually he was brought up here, and Helmut worked here until his death. He also lived almost forever, and his work was related to that of Green, so they were at least a good fit. He studied fat metabolism. He discovered a kind of protein called an iron sulfur protein, which came to be extremely important, and he developed a technique for studying metals in enzymes that made him world famous. Gobind Khurana was the fourth one that came to the Enzyme Institute. He grew up in India, poor family, somehow managed through his excellence to get educated, ended up at the University of British Columbia, where Henry Lardy got acquainted with him and recognized that he was a brilliant and very promising scientist in a place that wasn't going to nourish him as well as we could. So they actually went after him. Gobind decided to join the faculty here. He had spent his whole life learning how to chemically synthesize DNA and RNA. And this was at a time when the genetic code, how RNA is translated into protein, was not broken yet. We didn't have that answer. And what was lacking, we had the machinery that makes proteins, ribosome, transfer RNAs, energy, ATP, but we didn't have the nucleic acid to program the formation of a protein, so the ribosomes didn't do anything. Gobind Khurana knew how to make DNA pieces of modest length, and he made pieces of different sequence, and they used them one at a time to program the ribosome to make proteins. And depending on which sequence they programmed with, they got a different protein. And this was the key to the genetic code, and Gobind won or shared the Nobel Prize with Bob Holly and Marshall Nernberg. Masayasu Nomura came a little bit after Gobind Khurana, but they were contemporaries for some time. He was a genius at organizing scientists. He had, I think, at one time 30 postdocs in his lab. And he tackled what was an incredibly challenging problem, the structure and the assembly of the ribosomes themselves. They have dozens of proteins in them. And Masayasu used a combination of genetics and biochemistry to figure out how this multi-subunit thing is put together one step at a time. First you had to have this subunit, then you had to add this one, and then you could do this. It was a tour de force and made Nomura very prominent. And here's Mo Cleland. Mo was the most recent of the people in the Enzyme Institute to die. He died from a tragic accident in his own driveway when he slipped in the ice. Mo was a world-famous kineticist. He was very quantitatively inclined, and he used the speed at which a reaction occurs to measure the nature of the reaction. And he did so for lots of different enzymes, and he helped lots of different people do it for their enzyme, and became widely known for it, very widely known. Mo never liked me, but I always admired him. So this is the bottom line on the Institute for Enzyme Research. The people who came there were all really of one generation, almost. These people here. And the people who came there stayed there for a long time. So Lardy died, he was 93. Meinert was close to 90. They were probably licking their fingers of some of the stuff that they were making in the lab, and it added 20 years of their lives. But the sad truth here is that what we see is the birth of a place, a wonderful place, the Enzyme Institute, its development by people who cared a lot and were very good. And then, though it's not shown here, and I haven't told you about this, it eventually went downhill in terms of an institute and became more and more like a department. The campus needed the help of, for example, Enzyme Institute people to teach a course, and so that perk was lost. And slowly but surely, the Enzyme Institute quit being a special place and it became Biochemistry West. And finally, in 1999, the dean closed the door on it. There was lots of shimphing, but that was the end of the Enzyme Institute. It's still a great idea, and there are still a few places in the world, in fact, a few places here, that still operate on this principle. But most universities can't, because this way of doing science is much too expensive for most places to be able to afford. Questions? Yes.
What's the 2012 referring to in the Enzyme Institute?
1999 was when the dean said it's over. 2012 is when the last member of the Enzyme Institute actually moved out. The dean let them stay until they retired.
The dean of what college said in 1999?
This was not a dean of the college. This was the dean of the graduate school because the Enzyme Institute was not in a college.
Okay.
It was driven by the Enzyme Institute, the graduate school. So the dean of the graduate school was their boss.
Okay.
Yes.
I have a question about Warburg's study in 1930s. When he tried to measure the rate of the respiration, did he measure the intermediates present in the mixture or measure the volume of the oxygen directly?
This was in 1930 or 35, and he was using manometry, that is gasometric measurements.
So there was a way to measure the oxygen consumed directly with the...
Yes, when oxygen was consumed from the closed system, the manometer moved up to take up the space that the oxygen had taken. And it was an amazingly crude tool, but he did amazing good things with it. They all did.
Which dean was it who got rid of it?
You know the woman.
Oh, yes, of course.
Ginger. There were terrible hard feelings about this, and there was quite a fuss over it.
Yeah.
Yes.
Did I hear you say, when you were talking about the demise of the enzyme research and how so few institutes in the world have that operate on that kind of a model because of the costliness of it? Did I hear you say there are actually a few places on campus that still operate?
No, not on campus, but in the country.
Okay.
So,
Like what?
Cold Spring Harbor Laboratory in New York
Okay.
is such a place.
Right.
There is an institute in Seattle. I don't know the name of it, but it's a medical institute, and it stands alone. There is in Worcester. I don't know why it happened to be there, but in Worcester there is a cell biology institute with a small number of people who are their own bosses, basically. And I'm sure if I looked hard, I could find a few more. But it's hard to justify this because there are no students being trained. And for a university to take on major responsibilities with no students being trained is a hard thing.
I have another question.
Mm-hmm.
When you showed us the PowerPoint slide of the Kaiser Wilhelm Institute after it was bombed,
Mm-hmm.
do you know if that was a strategic bombing or just part of a big...
This was in Berlin.
Okay, so it was just...
Everything.
Maybe not particularly looking to...
No, I'm relatively sure that they didn't, but they couldn't aim bombs like they can now. Yeah. Okay. See you all on Thursday.
Thank you very much.
You're welcome.